It's a protein that you find in a jellyfish called Aquarium Victoria. And when you put blue light or UV light, it fluoresces green. And this is the picture of the jellyfish on your left. And what the fluorescence comes from specific spots that are on the jellyfish. So on the jellyfish, sort of around there, it's called the bell ring. And a bit about the history of GFP. In the jellyfish, the GFP has always been there for about 160 million years. But it wasn't until 1962 that it was isolated by a Japanese scientist called Osamu Shimamura. And in 1979, we characterized the structure of the fluorophore. And the fluorophore is the part of GFP that causes it to fluorescence. And in 1992, another scientist called he cloned and provided us with the nucleotide sequence of the wild type of GFP. And 2 years later, Martin Chalfie's lab, they expressed the coding sequence of GFP in E. coli, and then they also did it in a worm called C. elegans. And another 2 years later, we got the crystal structure. And in 1997, Roger Tien and his lab, they described a technique called FRET. It's Foster resonance energy transfer, and I'll tell you a bit more about it in the coming slides. So these are the guys that won the Nobel Prize. At the top is Osamu, and Martin in the middle, and Roger at the bottom. And their roles were really important, each one of them. Osamu was the first person to actually isolate and find the GFP in the jellyfish. And Martin Chalfie showed us how we could express GFP inside cells and be able to sort of see them glowing. And Roger Tien sort of gave us our general understanding of how GFP works, and he added to the color palette as well. And the way that GFP works is, first if you look at the structure, you can see it's sort of like a can. We call it a beta barrel, and the part that fluorescence is in the middle, so it's buried deep within the can. And so it's in the middle it fluoresces, and you've got the can structure around it. So you can imagine it to be like a light bulb, how a light bulb works. And it's the GFP was found with a companion protein called aquarin. And what aquarin does is it requires calcium, and it emits blue light, and the blue light is absorbed by GFP. And when the electrons inside the fluorophore are excited, they will then drop down to the ground state and emit green light while they do that. And in order for us to be able to really get to use GFP, we needed to expand the colors that we could use. And as you can see, there's a lot of colors that we've got. We've got some blue, we've got cyan, and now we have sort of enhanced colors so that you can really see what's going on inside the cell. Got some interesting names like M cherry, M strawberry, and M grape, and M raspberry as well. And this was all done by Roger Tien. So when you've got your GFP and you want to look at a protein inside a cell and see what it's doing, what you need to do first is find out what the gene of your protein that you're looking for is. And downstream of that, the coding sequence of your gene, you put in the GFP gene sequence onto it. Usually we put it just before the stop codon, but you can put it anywhere you want, so long as it doesn't affect how the protein works. And once you put in your protein in the reading frame of your, so if you put the GFP gene in the reading frame of your protein, when you translate the protein and the protein folds, you end up with a fusion which contains your protein that you're interested in and GFP, and wherever that protein goes inside the cell, you can see, because GFP sort of lighted that up. And so what we're using it for so far, in this picture, you've got the oocyte of the mouse, and you can see the actin. You can't see it very well because it's not dark enough, but you can see the actin cytoskeleton is labeled with green, and you can see the chromosomes are labeled red. Yeah, so you can sort of have a look there. So we've used it a lot, and we'll continue to use it. And someone kind of came up with this quote that humans has found so many uses for GFP that it would astound a jellyfish, which I thought was quite funny. And if you look on Google Scholar and you're searching for scientific articles, you get more than 1.1 million articles. And if you've heard about Alba, the glowing rabbit, it's also another animal that's been used for research, and it's expressing GFP. And we've got a lot of animals, including monkeys, pigs, rabbits, mice, fish, flies, and many more. And most of you may have heard about the Green Hawk, at least the story. The directors of the film decided to explain how he's green by saying it's GFP. But of course, if it was GFP, then you would need to shine UV light on him to see him as green. So it's a way for them to sort of use it, but it's not really true. We also use GFP in biosensors. So I'm assuming right now you're probably learning a lot about the heart and how you require calcium and so on. And if you want to find out the movement of calcium, you can also use GFP as well. And we use another technique called FRAP, and this is to study how a protein is moving around in a cell. So if you photo bleach a small area, that particular area will lose fluorescence, and then the proteins that are still fluorescing will come and repopulate that area, and so you can work out the time of sort of movement from one place to another within the cell. And in another technique called FRET, you label two proteins with two different fluorophores, and if you can think of the emission spectrum of one being different to the other one, but they're overlapping, so that when you shine light and you excite the first fluorophore, the second one will only be able to sort of shine the different light if they are close, if the proteins are close. And this is a nice picture of a cancer. I don't know if you can see that that's a rat. I can't see it very well. On the right, it's a rat that has a brain tumor inside of it. It's expressing a different fluorescent protein called DS red, so it's a red fluorescent protein. And in that rat, the tumor is red, and the blood vessels around the tumor are green, so it kind of appears yellow, but they're green. And we've used GFP to study how cancer moves around the body, but we've used it in animals so far, and we've also used to study how it grows and so on, and we can do drug evaluations as well. And this is a picture of neurons in the brain that are all labeled in different color, and it's a technique called the rainbow. And since it's really difficult to study the brain, we've started now by labeling each single neuron, and we can look at the connections that are made so that we can learn more about how the brain functions, and we can get some nice pictures as well. And And we've used it as well to learn about a new thing about HIV that we didn't know before. T cells are cells of the immune system that help fight the infection when you've got HIV. But of course, they are the cells that are infected by HIV. And you can see the green one is the HIV proteins, and the red is the T cell that's not infected by HIV. And this is sort of a time scale, so it starts at time 0 and time 65 seconds later. And you can see that on the uninfected T cell, the infected T cell, they form like a little strand, like a little sticky strand that connects two cells together. And the HIV proteins are able to actually move between those little sticky strands, and so they can infect another T cells without actually touching it, but without physical contact between the two T cells. So that's another really interesting way. And so in the future, we've recognized that GFP has been revolutionary, and we're looking to improve certain colors like the orange, the red, and the infrared. The reason is they would be more useful to examine thick specimen, because if you shine red light within something, you can see it better because it's more transparent to the skin. And another second area for future growth is using fluorescent-based measurements as well. And so this is all for my presentation. I hope that you enjoyed it and you found it useful in the future. And if you want to fill in the feedback form, just let me know how you found it. Thank you very much. If you have any questions, I'm happy to answer them. And if you want more, I have references.